As Natalia said kindly, I did my PhD at Eawag in Zurich, and I worked a lot on trying to quantify AMR in wastewater. And today I will give you a broad overview about what I've been doing and some of the next steps that I, and not only me, believe should be taken to improve the relevance of wastewater-based surveillance for AMR. So I guess that you all already know what is antimicrobial resistance, namely the ability of microorganisms to inhibit antimicrobials. So if a person gets infected with a bacterium which is resistant to an antibiotic, then this patient can experience a longer morbidity, longer hospital stay with associated cost, and a higher risk of mortality. And so only in 2000, the failure to address the problem might lead up to 10 million deaths per year by 2050 worldwide and associated cost equivalent to $60 trillion per year. And that has been estimated in 2016. And just to give you a comparison, nowadays the major cause of deaths are cancer, which amounts to 8.2 million deaths per year. 1.5 died due to diabetes and 1.2 due to road accidents. So this 10 millions is really significant. And only in 2019, bacterial antimicrobial resistance was responsible for almost 1.3 million deaths worldwide. And if we zoom in a little bit, we also know that only in the United States, every year there are 2 million infections that are caused by resistant bacteria. Unfortunately, 25,000 people die every year due to resistant infections, both in the US and in Europe. And in low resource settings, this burden is even higher. And so it has been estimated that in India, for example, one child dies every 9 minutes due to a resistant infection caused by a resistant bacteria. So to tackle antimicrobial resistance, we need to monitor it. And the main limitation is that antimicrobial resistance surveillance nowadays is driven by clinical data, which is great, but has limitations. So first of all, there is a lack of representativeness. So this means that only people that have access to healthcare and go to hospitals are actually screened for resistance. And we have no idea how many of us, maybe in this moment, are carrying resistant bacteria and we just don't know it. There is a lot of resources needed in order to diagnose antimicrobial resistance and to screen for it in clinical settings, which requires a lot of money, a lot of time, and a lot of people. And last but not least, there is the problem and the limitation of underreporting. So not everyone reports about antimicrobial resistance to public health authorities. And even in countries like Switzerland, where we have a very well-established clinical surveillance system for AMR, not everyone is obliged to report AMR cases. And so, in addition to all of it, we also know that antimicrobial resistance is not a problem limited to the clinical settings, but AMR is a One Health problem, namely that resistant bacteria and resistant genes, they do spread among different compartments, namely from the clinics to the community, households, animal, the environment, rivers, waters. We can get resistance through traveling, through the food chain, depending on what we eat, and we can spread it up to our pets or to our friends or to the people we encounter. And in this very nice diagram, we also see that one of the transmission pathway is through wastewater. And this is because we do shed antimicrobial resistant genes and antibiotic resistant bacteria into the wastewater. And sometimes, unfortunately, these resistant genes are also released again into the environment after treatment. So when I started my PhD 5 years ago, as Natalia said, at the Eawag, we were already collecting wastewater because we were quantifying SARS-CoV-2 during the pandemic, and that worked perfectly. And so one day I was like, you know what? We can also quantify AMR. So I wasn't specific. I was just, okay, we can quantify AMR because if you use wastewater, this means that everyone connected to that sewer sheds into this pipe and sheds into this system. And so what you collect at the inflow before the wastewater treatment plants should be representative of what the whole community is shedding, right? And so it doesn't depend from health-seeking behavior or going to the hospital or being sick. And so I was very excited about that. But then with time, the more I was trying to do it, and I was not the only one, we just realized that antimicrobial resistant data from wastewater are not the same as those that we obtain from the clinical settings. And what we obtain from the clinics is what is usually nowadays used to inform antibiotic stewardship policy. And so there were some concerns about how useful this wastewater data about AMR can be for health decision making and policy. And so 1 year ago, 1.5 years ago, I was at a conference, the Environmental Dimension of Antimicrobial Resistance Conference in Montreal, Canada. And together with many experts of the field, we performed a workshop aiming at really trying to understand how we can make wastewater surveillance for antimicrobial resistance clinically relevant. And so we came up with this short viewpoint, which answer 4 questions that we believed were important to answer. And so, in order to make wastewater-based surveillance of AMR clinic policy relevant, we need to know what there is already out there nowadays that is used to drive antimicrobial stewardship policy. And so we agreed that one of the sectors that provide useful indicators is the clinical setting. And so there we track resistant trends in the infection. So we have the trends of resistant infections and not of resistant bacteria, which allow clinicians to develop antibiograms and to guide antibiotic prescriptions, for example. But we also have indicator coming from the agricultural sector. And so in this sector, we mainly focus on, not we, but people mainly focus on monitoring antibiotic consumption. And so thanks to that, we can identify areas of high selective pressure because more antibiotics have been used. But it's also useful to monitor the transmission pathway of antibiotics and resistant genes through the food chain and through zoonotic pathways as well. And lastly, there are indicators that come from the environmental sector. So there are a lot of studies, especially research-oriented studies, that aim at investigating antimicrobial resistance spreads over time and over space. But one of the main indicators coming from the environmental sector is that it guides wastewater management. So imagine when we quantify AMR in the river or in the lakes of Switzerland, and these might help us understand if our wastewater treatment is enough or not. So these are the indicators that are already out there, but then we wanted to try to come up with what can actually wastewater-based surveillance provide us. So which type of indicator, which type of data can we collect? And this is a very important point for me, and for today as well, because I believe that it really depends from your objective. So it really depends from what do you want to find out from wastewater, because you can find a lot of things related to AMR, and depending on what is your objective and what your targets and what you measure, then you will use a different methods. But on one thing we agreed. So whatever we are going to measure, it should be clinically relevant. And this is because we are looking for indicators that could guide antibiotic stewardship. And this is because we want to decrease the burden of AMR on healthcare settings or on the number of deaths that can occur every year due to AMR. And so it needs to be clinically relevant, and it should be low prevalent. So this means that we don't want to monitor something that we know it's already out there in the pipes naturally, because otherwise we don't really know if what we are measuring comes really from the clinics, from the people, or from the animals, or it's just out there because it's part of the environment itself. And so What can we measure? So we can measure antibiotic resistant bacteria themselves, so we can grow them and count them. But we can also quantify the concentration of resistant genes or of antibiotics. We can investigate mobile genetic elements to understand how resistance spread. And depending on what we want to measure, we can use different approaches. So if we want to measure and enumerate bacteria themselves, then we need to grow them using culture-based technique. If we want to quantify the concentration of resistant genes, then we could use qPCR, dPCR, so target amplification. But if we just want to know whatever there is out there, everything, then we could use metagenomic. And so we just sequence whatever there is into the wastewater, and maybe we could find out newly emerging antibiotic resistant genes. And so, depending on what you measure, you use different methods. But then, where do you measure? It depends from the setting. So, if you have a centralized wastewater treatment system, like in Switzerland, then it makes sense to collect at the wastewater treatment plants because you assume that is representative of the whole community connected to it. However, in some settings, we don't have centralized system, but we have decentralized wastewater treatment. And so in these settings, how can we use wastewater-based surveillance? And in that way, we could target specific communities instead of the whole community. So, for example, schools, hospitals, universities, and so on. And so this was the second question, which was like, okay, what we can get out from wastewater theoretically. Now, the third question was, okay, we know what there is already out there and which is used for antimicrobial stewardship policy. We know what wastewater could potentially provide us. How do we integrate wastewater with what is already out there in order to better understand the epidemiology and the spread of AMR? And so first of all, we need to integrate our data with clinical surveillance. So we want to know, as I said, what is clinically relevant. And so clinical surveillance needs to be there because we want to know what we can measure in wastewater that is actually relevant for policy. But then we also need to integrate the wastewater-based surveillance with the environment and with the agricultural sector. So, for example, imagine you are quantifying bacteria into the wastewater and you are not really sure if what you get from the wastewater really comes from the community. Maybe it comes from the environment. And so one approach would be to perform whole genome sequencing and to compare the DNAs of bacteria coming from different sectors to see if those in the wastewater are very similar to those in the clinical settings. But we also want to know the antibiotic consumption that is going on in these different sectors, especially if we are going to measure antibiotics into the wastewater. And then we need a huge investment, and not only financial, but we need to have a commitment and a continuity coming from institution, from people, from researcher, and from people that actually also fund the research. I mean, clinical surveillance is out there since decades now, and so we clearly know what can we do with it, but we are not really sure yet what we can do with wastewater-based surveillance. And so I believe we do need some time series and some continuity before to have a better overview of what could how could be helpful. And then, very important, we need a strong collaboration. So all the researchers that are still trying to make wastewater-based surveillance for AMR clinically relevant, they should highly collaborate with clinicians, with stakeholders involved in antimicrobial stewardship, with the communities, with the veterinarians, with other researchers worldwide. And so this is really a very important point. And now I would like to provide you some examples of which type of data we have been able to obtain through our wastewater monitoring at Eawag. And so I'll just give you a broad overview, but I hope that it allows you to see a little bit more what can be obtained. So we performed a longitudinal surveillance of AMR in Swiss wastewater. You see on the top left the Swiss map with 6 wastewater treatment plants highlighted. And so we started in November 2021, and this is still ongoing. So people are still doing this procedure. 6 wastewater treatment plants, 1 to 4 times a month. So at the beginning, we quantify AMR in wastewater weekly, and now, for example, is only monthly. And we targeted three different bacteria, which are highly clinically relevant and listed among the priority pathogen list provided by WHO, which are ESBL E. coli, so Escherichia coli that are resistant to broad spectrum antibiotics, but also carbapenem resistant E. coli and vancomycin resistant enterococcus species. So what we did was to collect wastewater and to perform culture-based enumeration. So we are enumerating the bacteria themselves and not the resistant genes. So we spread some wastewater on these agar plates, as you can see on the bottom left. And for each time point and location and for each target, we have two different plates, one with antibiotic and one without antibiotic. And on the one with antibiotic, only resistant bacteria grow, while in the one without, all of them grow. And so once we have these two things, we can count the number of resistant over total bacteria, and we can obtain a percentage of ESBL E. coli over total E. coli or of VRE over total Enterococcus species. Another thing we did was to isolate some of these bacteria, resistant bacteria from wastewater at different time points, culture them, and perform whole genome sequencing, and so sequence their DNA. And these would have allowed us to see if what we observe in wastewater would come from the clinics or would circulate in the clinics as well, or would be just in the environment, in animals, and so on. And so the first study I would like to present you is very recent. So it should, we just submitted it last week, and it was a collaboration with ANRESIS, which is the AMR surveillance network in Switzerland. And so how did we integrate our wastewater data with clinical surveillance? Because as I said before, this is one of the most important points to make wastewater-based surveillance really relevant at policy level. And so we have the community and we have wastewater. And wastewater actually should mirror what is circulating in the community. As part of the community, we have people that carry susceptible bacteria in green and resistant bacteria in red. And within the community, we have the clinical compartment. But in there, we also have susceptible and resistant bacteria. And so once one person gets into the clinic with a bacterial infection, they are treated with antibiotics. And these antibiotics can either treat the infection, but at the same time, they could also cause selective pressure on other bacteria species and increase resistance theoretically, right? And so to integrate our wastewater data, we also retrieved data from ANRESIS, as I said, about the percentage of human infections that are resistant and also about antibiotic consumption data in humans over the same time period. And so the first thing we observed here is like the timeline of the resistance of ESBL E. coli over total E. coli in wastewater over 3 years. And we observed that there was an increase of 0.5% every year. And wastewater surveillance of antibiotic-resistant bacteria reflects actually long-term clinical trends because we observed the exact same increase on a long term also from clinical data. However, as you can see here, the two lines are not completely overlapping because this indicates that on a short term, there is not really a correlation, and I'm not showing you now. But on a long term, what we observe in the wastewater represents what we observe in the clinics as well. And this pattern has also been observed for the other two pathogens, even if slightly different. So for carbapenemase-resistant E. coli on the left, we observe an increase in the clinical data of 0.04%, but it remains stable in wastewater. And for VRE, we observed a higher increase into the clinical data compared to the wastewater in blue. And so this means actually that the percentage of resistant infections increases faster compared to the percentage of resistant bacteria into the wastewater. Another thing that we observed, just to contextualize wastewater resistance, is that the rank order, namely of these three pathogens, the percentage of ESBL E. coli is higher than the one of VRE and then the one of carbapenemase resistant E. coli in both compartments. But we also observed that the percentage of resistant infections in red coming from the clinics is higher than the percentage of resistant bacteria. And this is because these are essentially two different measures, but this could be explained by two reasons. The first one is that we observe more resistant infections, which are more infections that are resistant than susceptible because susceptible bacteria cause fewer or milder infections and are therefore undersampled. Or the other reason might be that resistant bacteria might be more likely to cause infections that require medical care, and so that's why they are oversampled in the clinical setting. Then we also retrieved clinical antibiotic consumption data. And we divided antibiotics in two groups for each target. So we have antibiotics that select for that specific target resistance and antibiotics that are treated that are used to treat that specific target. So let's take ESBL E. coli again here in blue, the percentage into the wastewater. And then we have the consumption of fourth generation cephalosporin over the same time period. And fourth generation cephalosporin is exactly what this type of ESBL E. coli is resistant to. And here we didn't observe any association, namely that an increase in the consumption of these antibiotic over our study period didn't show an increase of ESBL E. coli, potentially then indicating that actually this antibiotic consumption select for resistance of ESBL E. coli. The other category of antibiotics was for ESBL E. coli, what is being used to treat these ESBL E. coli infections, and in that case would be carbapenem. And again, we didn't observe any association, meaning that an increase in carbapenem consumption is not associated with an increase in ESBL E. coli in wastewater. So there is an increase of ESBL E. coli, even if we don't observe an increase in antibiotic that is used to treat them. And so this pattern is maintained also for the other targets, and I'm not going to lose too much time about the details. The main point here is like that the short-term prescribing pattern of this antibiotic do not directly translate to wastewater signals. So this study allows us to understand that actually wastewater and clinical on a long term do match, but wastewater has no correlation with antibiotic consumption. Another question that we could retrieve from wastewater surveillance in Switzerland was to try to understand if wastewater can provide insights into the prevalence of ESBL carriage in the population. And so, in other words, can we estimate the number of people that carry ESBL E. coli in Switzerland, starting from a percentage that we observe in wastewater? And so we assume that the percentage of people that carry ESBL E. coli, with these red hats here, depends on the percentage of ESBL E. coli that we measure into the wastewater and the percentage of ESBL E. coli out of total E. coli in the gut of the carriers. And so I hope you're following me on this, but we found a 1.9% ESBL E. coli in wastewater, right? So if we assume that the people that carry ESBL E. coli in their gut have also 1.9%, then this means that 100% of people carry ESBL E. coli in Switzerland. However, the higher the percentage in the gut of the carrier, the lower the percentage of people that are responsible for shedding this 1.9% into the wastewater that we found. So if we assume that people that carry ESBL E. coli have 19% of ESBL out of total E. coli in their gut, then this means that in Switzerland, based on our 1.9% observed in wastewater, only 10% of people actually carry ESBL E. coli. As you see, here we could choose different percentages of ESBL E. coli in the gut of the carrier. And with this study, we kind of understood that it would be possible to use wastewater-based surveillance as an indicator of carriage at the population level, but we do need cohort studies telling us how much carrier gut actually have ESBL E. coli out of total E. coli. What is the percentage of it? And so again, this is another example of how we could use wastewater surveillance, but again, it needs to be integrated in a bigger framework, namely be integrated with clinical or other type of studies. And now I'd like to briefly mention you other type of data that we can retrieve from wastewater, which is the number of transmissions, or namely how often these resistant bacteria, here in red, do travel among these different compartments and how often are they transmitted. And this is something that we also try to do using wastewater bacteria, as I told you before. And so, very briefly on the methodology, what we did was to whole genome sequence 800 ESBL E. coli. And so we collected them from wastewater, as I showed you before, but we also collected them from different sources, namely from the clinical settings, from wildlife, from the environment, livestock, agriculture. We sequenced their DNA. We built phylogenies, which aimed at understanding how related these bacteria were. And then we performed this ancestral state reconstruction, which essentially what you need to know is that it allowed us to double check how intermixed the bacteria that come from wastewater, for example, are with those that come from the clinics. In other words, what we sample in wastewater, is it clinically relevant? Is it something that is circulating in humans as well or not? And these DNAs are completely different. And so I go directly to the point, and I guide you through this graph now. We have in blue here these two box plots, which show the number of transitions of exchange based on this risk construction among all clinical and wastewater isolates that we had. And we see that the number of transitions, so the exchange, is significantly less than what we expect. And this means that wastewater and clinics, in this regard, for all these bacteria, are actually two different compartments. And so this suggests that what we find in wastewater is not really circulating into the clinics. However, on the right side, we have subsets of our ESBL E. coli, and each color represents a clinically relevant E. coli strain, so those that are actually causing the majority of infection for ESBL E. coli, and so that they are associated with people and are not just out there in the environment. And we see that if we consider only these subsets, we observe that there is exchange between wastewater and the clinical settings. And so this means that what we find into the wastewater captures community circulation, because these are community circulating strains, which in addition are also clinically relevant. And so we are very happy with that. And now, to conclude, I would like to bring up again the last and fourth questions of our viewpoint that we generated after this workshop at the conference. And so how can now the information that we derived from wastewater contribute to the formulation of effective public health policies or interventions? And I'm sorry to disappoint you here, but I don't have a precise answer. What I hope that you understand and you enjoy it is that one of the most important thing about wastewater-based surveillance for AMR is to bridge the gap between clinical surveillance and environmental or agricultural surveillance. So only through this bridging we can better collaborate with all the people involved in antimicrobial resistance surveillance, with all the stakeholders across One Health sectors, and we should better be able to integrate wastewater-based surveillance in current surveillance programs, because then we could better compare and we would be better aware of what's going on in wastewater as well, which would increase the awareness and in the community, but also in scientists and in policymakers, and hopefully we could enhance the public support of wastewater-based surveillance for AMR. And so with this slide, I concluded my talk. And so I thank you a lot for listening. And